Welcome to Sanctuary Soundscapes. My name is Jody Patterson, Director for Gray's Reef National Marine Sanctuary Foundation. Thank you for joining us as citizen scientists and volunteers to help support this research project to better understand the soundscapes of our National Marine Sanctuary. If you will, join us on our website, graysreefnmsf.org, under the Get Involved tab for the Citizen Science Project. You'll find additional resources and inspiration to help prepare you to support the Sanctuary Soundscape Project, including all of our project frequently asked questions and education resources to put this entire project in greater perspective. NOAA has created a number of different resources to help introduce ocean acoustics and this particular study using passive acoustic data to monitor ships and ocean life. So please take some time, visit some of these NOAA resources, and learn much more about ocean acoustics. And now we'll introduce you to some of the processes and procedures for participation in the Citizen Science Project. So please follow along and feel free to ask any questions at graysreef@marinesanctuary.org. The passive acoustic data deployments in the SanctSound projects are denoted by these orange circles around the sanctuaries that are participating in this project. We're going to click on right here off the coast of Georgia for the Gray's Reef project files. And as you can see, there are 4 folders here, and we can request these data either directly through NCEI, but please note that there are almost 1900 gigabytes of files that are available to be downloaded. So unless you have that kind of space on your computer, we suggest that you request access from Google. And as we do that here, it may take a few seconds for this screen to load up. This is a large amount of data that is archived onto this cloud platform, but this will enable us to just select individual files as opposed to downloading the entire database. And as we can see here, we're now in Google's cloud platform for NOAA passive bioacoustics. Within this platform, we see a number of different folders here from some of the different projects that they are focusing on, and again, we want the SanctSound project. So we'll click that folder, and here we will see a number of other folders, each referencing different research areas in this project. So while it doesn't directly give us that Gray's Reef data, we can find Gray's Reef 01, GR02, and GR03. These are the file folders that we will be using for this Citizen Science Project. So to begin with, let's start with GR01. When you are working on your projects, you will be assigned a folder or files to reference. Those files are the ones that you will work through and to help catalog the soundscapes and then report that information back to us. And so what we will do now is show an example of how to download a file, and then we will go through the processes for analyzing that file. So once we are in the GR01 file, it will give us 3 additional choices here. We would like the audio data. So go ahead and click into audio, and it will bring up those files for us to choose from. And we will omit this first file that is an unusually small size here. This was a post-deployment audio, and what we would like to see are all of the in-situ audio files, and there are quite a few. I'll just give you a quick rundown of some of these files, and if you'll note, each one of them is 1.9 gigabyte. That is substantial memory to take up on your computer, so please make sure that you have that either available on an external drive or on your desktop or your laptop available. And what I would recommend is that as you work through each file, maybe go back and delete previous files that you no longer need, just to give yourself enough space on your computer memory. But what you'll note here is this is one page of 50 files, and there are 569 pages of files just within this Grays Reef 1 folder. So as you can see, we have many hours of recordings to listen to. And that is where you are going to come into play. You will help us by providing ears and eyes to listen and see what is on all of these data files, and help us catalog that collection so that we have a better sense of the sanctuary soundscape. So let's begin here. We'll start with this very first file, and we would like to select it so that you can take a little bit of a closer look at that, and you can even preview this file if you would like to. So we could play this file here if we wanted to sit here and listen to about 5.5 hours of this acoustic file. You're welcome to do that. But that's not the purpose of this project. What we'll need to do is download this file. So step 1, download the file. We're doing that now, and then I'll come right back and we'll take a look at Raven Light software. Okay, so the software that we'll be using to help us analyze the acoustic data from the Sanctuary Soundscapes project is called Raven Light, produced by the Cornell Lab Center for Conservation Bioacoustics. And this is free software that lets users record, save, and visualize sounds as spectrograms and waveforms. And while it doesn't have all the upgraded features of the Raven Pro software, it will do everything that we need to do in terms of annotating the content in that acoustic data. So what you'll want to do is go ahead and download that Raven Light software. And of course, it will try and sell you on the Raven Pro with all of their features, but for our purposes, Raven Light will be just fine. So if you want to, go ahead and scroll to the bottom of that page and download Raven Light. It will offer you a free license, which, go ahead and grab that if it's free. So then you'll just need to follow the requirements for downloading Raven Light. If you select Download Now, select the version that you're looking for, go ahead and download and get the free license while you're at it. That will take a few minutes to download that software, but go ahead and get that going, and we'll be back to show you how to use that software once it is installed. Once you have Raven Light installed onto your computer, go ahead and launch that program. Upon launching, you'll see a screen that looks like this. What we'll need to do now is open one of our files. So come over here to the upper left-hand corner and drop down Open Sound Files. Now you should have the data file that you downloaded from the Grays Reef 01 file somewhere handy in a folder that you can easily find. Go ahead and find that file, select it, and open. It will ask you to configure a new sound window. At this point in time, I suggest to leave these presets at their default. You will be able to learn more about these presets and what they relate to as you learn more about analyzing the acoustic audio. In the meantime, leave these at their default settings and just select OK. It will take maybe a few seconds or a couple of minutes to open this, just depending on the processing power of your computer. But as it processes and opens this file, it will first open a waveform and then a spectrogram view. So the waveform is being created, or has been created, and now the spectrogram is computing. So we'll just give that a few seconds to finish processing. Okay, now with our audio file open in our Ravenlight window, let's go ahead and unclick this waveform image here at the top of our viewing window. We have two options here, waveform and spectrogram. Most of our observations will be done with the spectrogram view, so we can go ahead and unclick the waveform. What I also like to do is change the colors of my spectrogram, because I like a little bit of a cooler image, but you are of course welcome to use any of these color palettes that you like that can change the way that you view your spectrogram. It's all a preference, so just choose whichever you'd like. And so what we want to do is begin to visualize our acoustic audio in the spectrogram and be able to pick out some of the key signatures of the audio just by visually scanning over it. And as you can see, the color of your audio or your spectrogram file will really help delineate some of those signatures from just some of the broadband spectrogram of noise that you see here. Most of the acoustic audio recorded at Gray's Reef is within this broadband range here between, say, 4 kilohertz and 14 kilohertz. Within this range here, most of that noise or sound from our sanctuary occurs. But we'll drill down a little bit closer, look at the recording over time, and dig into the frequency range, which is our Y. On our x-axis is time, on our y-axis is the frequency of sound. So we'll drill in a little bit closer into time and into that frequency range to better identify some of the acoustic signatures. So let's go ahead and play, and I will be highlighting for you some of the regular standard tools that you'll want to use for this observation process. And I will show you how to annotate your files as we observe and listen to them. So what you'll notice right off are all the snapping shrimp that are at Gray's Reef. The little popping of bubbles from those snapping shrimp produce a tremendous sound that you'll hear throughout the acoustic soundscapes recording at Gray's Reef. Again, in this broadband range, a lot of that sound is found. So we won't necessarily need you to annotate snapping shrimp in our data set because it's pretty much always there. So that's not so much of a need to know, but it's some of the other animals that we are less familiar with in terms of the sounds or the noises that they make. So why don't we look at that a little bit closer in our data. And what we can do here, we have a couple of magnifiers here. This magnifier will zoom in on our x-axis, which is in time, and these two magnifiers will zoom into our frequency. So why don't we go ahead and zoom into our x-axis, which will bring us a little bit closer in time to this data set. So you can see here at the bottom, it's beginning to broaden out. It was a little over 5 hours in recording time, and we're now showing just under an hour. So we can zoom in a little closer and closer, and the closer we go, you'll begin to see the screen actually start to scroll with the time. So we can get down to the minutia of this data. And as you can see, it continues to scroll forward. If that's a little too close, we can of course zoom out so that we can see that broader range. And likewise with our frequency range, we can zoom in. And as you see, it's zooming down to the lower frequency range, down into the hertz, and up into the kilohertz. And then we can also scroll through that with these tools here. So if we want to zoom out just a wee bit of that frequency range, you can just take a look and see some of the delineation. Really all we hear right now are those snapping shrimp, but here in a moment it gets much livelier. Zoom out on our time scale just a little bit and see what's coming. If we look down here in the lower frequency range, we can see some small bursts here and there. Those are often fish grunts, pops, or some animal brushing against the hydrophone. Usually it's something that you won't be able to identify, but you might want to call it something in particular if it stands out. So let's zoom in over time again and see what we hear. What we can do here is we can pause this and stop the playback, and we can choose instead play scrolling, so that way it will continue to move the screen forward as it plays the audio. Nothing too interesting right now other than snapping shrimp. Let's look a little closer at the frequency, at these lower frequencies. There's a section here that I want to listen to. Let's see what it sounds like. You can grab and scroll your screen if it's moving a little too slowly. But these are just some little pops that are not quite the snapping shrimp. But if we want to stop this for a second, let's choose our selection tool and come down to the series of pops. We just want to click and drag and release, and then that will generate a selection table below. And what that is telling us is at this time it begins and at this time it ends. Within this lowest range of frequency and up to this highest range of frequency is when we observed it. And what we observed are popping. Now when we play again, some very light popping sounds. We just scroll up through the spectrum frequency and just take a look at what's coming. Not too much to hear in this clip. So why don't we grab it and pull it forward, see if we see anything more interesting. More popping. Sounds like something grazing right here. Right here. So let's go grab our selection tool. It sounded like munching. Play it again. Some of those are some very nondescript sounds. We're not quite sure what they are, but researchers may be interested in popping or munching sounds that are generated from the Soundscapes data. So by providing this annotation, they'll know to listen back for these areas of observations, and can further identify what those sounds may be. Now we see some sounds coming up in our data, and if we want to zoom a little bit closer to those, let's get a little bit closer here and take a listen. And we have some telltale dolphin calls. Now let's say we want to take a little bit closer of a look at this. Let's pause that, and we're zoomed in really to this lower frequency range in which we can see these dolphin calls start here. But let's zoom out on this. And as we do, we can see the signature of these dolphin calls extends from the lowest frequency range pretty high up into that spectrogram range here. So if we want, we can go ahead and grab our selection tool. Let's go ahead and annotate this, and we'll go all the way up. Try and get as much of that as we can within this range. And again, it tells us when it begins, when it ends, and the frequency range. And we have dolphins. We're not sure what they're doing, but they're communicating, so we'll just say dolphins. If we want to play that again, we have a few little sections of dolphin around this area here. So let's look at that a little bit closer. We'll look at it closer in over time. And when I do that, some of these signatures really begin to pop out in our data. So we've zoomed in over time, we're zoomed out on our frequency, and see those dolphin calls coming up. We don't even have to hear them yet. We know they're coming. Look at all the dolphins. We've got this whole section here of dolphin calls. Those are wonderful. Let's go back and let's try and grab all of that section. Look at all the dolphin calls here. So it starts around 12:19. Let's get our selection tool. Click and drag all the way up and over, just as far as we can see these signatures. We know there's a lot of dolphin activity in this area. Look at all these guys. And they keep going until, let's say about here. So let's stop here. And again, we'll just say dolphins. Let's play that again. We heard something thumping against the hydrophone there. Maybe a dolphin brushed up against it, swam by. Great. I love these guys. Okay, so you see we have a few different annotated observations here. This is just a dolphin-rich file. And so now you have a sense of where these acoustic signatures are in the data, where you can best identify them, then you can begin to zoom into those areas to look for them. So I see some patterns coming up here, so I want to scroll a little closer into these, see if we can get a better look at them. So I'm zooming in closer over time. Some interesting patterns that are emerging. And I'll also try and zoom into the frequency here, see if I can tease out some of the signature a little bit. Oh, that's a little too far. Zoom out. Let's play here. Some more dolphins and some echolocation. And we can come over here and scroll up. You can see the entire spectrogram in this view. But if this is going a little too quickly for you, zoom out over time, and that will slow that view down a little bit. And you can get a sense of what's coming. Some knocking and popping. This may be some type of fish movement around the hydrophone. You hear more of this munching sound in this lower frequency. Let's zoom into that. Can't be sure what that is without a video. But we can grab this again and look for interesting signatures in our spectrogram, and that's what you'll want to listen for. Not sure what that was. Sounds like a flutter. So we see 3 distinct pops right here. Let's go ahead and grab those. That may be some type of fish behavior. So we'll just say popping, because we don't know what it was. But a marine acoustician may be able to correctly identify that, or when we're able to pair a video camera with a hydrophone, then we can absolutely begin to ground truth what some of these sounds are. Okay, but some of those fish pops and grunts and general munching sounds are some of the minutiae of the soundscape data. The marine mammals, such as dolphins and whales that you hear, will have some of the more distinct calls and distinct signatures within this data. So I'll just give you again a better image of what that looks like as we a little bit more quickly scroll through some of this data. And as you can see, we have some distinct signatures beginning in the data around here. So why don't we take a listen to this time series. And as you can see at the very top of our frequency range, there's not a whole lot happening up here except for the occasional streak. This occasional streak we're going to hear. Let's listen. Not very clear. You'll hear it more clearly as this section arrives. There's some type of grunting, but I also hear dolphin echolocation. So begin to see more streaking in the upper layers of the frequency range here. A lot of that streaking is a result of the echolocation. Listen. I'm going to zoom out of the frequency range just a little bit so that you can see it coming. We can see the entire frequency spectrogram here, and we can see that something's coming. Just little fish pops. And if your assignment was fish pops and grunts, this is where you would focus, and this is what you would grab, these little pops. And again, down to our annotation table, popping. Okay. Continue on with that. All right, let's get to something more interesting. More of that munching sound. And we see, rather than hear, something else coming up with a much stronger visual. The dolphins are very noisy in this section. So we can see them long before we heard them. We saw that these dolphin signatures were in the data and these lower frequency ranges, which is where we can typically find most of our marine mammals, in these lower ranges. But this entire section could be annotated for dolphins. So let's go back to where we saw them start with their signature right around here. So let's grab our selection tool and grab this whole range of dolphin calls. Okay, so I can see it distinctly to about right here. So we'll say dolphins. Okay, and play again. Now, you're not expected to know what kind of dolphins these are, or even exactly what the behavior is that these dolphins are doing. The more information you can provide in these annotation tables, the better, especially the beginning and end of the time and the frequency range. That will help those acoustic researchers drill back into this data to find what they're looking for. So you're the front line as the citizen scientist to help with the observation and identification of our acoustic soundscape at Gray's Reef. So why don't we do this? Let's pause here, and I want you to take a broader look at some of this data, or rather from a bigger picture. So I'm zooming out over time. I was zoomed very far in to just let each minute scroll past us so that we could look at those signatures. But when I zoom out, I'd like to see the entire section of this acoustic data. And so there's something else that you can see in the data when you look at it over time. We see here at the bottom in this lower frequency range a section that begins to build and grow and then dissolves over time. And when we look at that, it takes a little bit of time to listen to this entire clip because it's almost the duration of an hour where this noise begins to build and eventually dissipates out of the acoustic audio. But we can go ahead and select where it looks like that sound or that noise is beginning, and we can just drill in a little bit closer to that. And I just want to make sure that I've got all the way to the lowest part. Okay, so when I select that, now I want to zoom to that selection because that's all I want to play right now is that selection. So when I zoom to that, it will bring up just that frequency range, and it will bring up just that selection of time that I've chosen. And what I want to do now is just play that. And when we look at that, here, let's zoom out of it so that we can see a little bit better what it is we're looking at in the data. So let's begin playing here. And it's very light. You can barely hear anything at this point. We can just begin to see it in the color map here in our spectrogram. And it's intensified here in this section. So we can zoom into this section a little bit more and see if we can hear a little more clearly what this noise is. And it's just a washout continuous sound that saturates this lower frequency range. And that sounds like an outboard engine to me, especially because of how it is reflecting in our data, being consistent over a long period of time, and begins to dissipate out of the area. My guess is it doesn't come very close to the hydrophone station. It's pretty remote, pretty distant, but we can still hear it. Sound travels at an incredible rate through water. Water helps sound travel faster than air, and as a result, any sound, any noise in the waterway will be carried many, many miles. So here I'll go ahead and annotate this as outboard engine. We'll zoom out on that again. And again, just look at it within our Soundscape data. Okay. More of those dolphins. Okay, so we have a few different selections that are noted within our selection table, including popping and munching, dolphins, more popping, more dolphins, more outboard engine. So we have 8 different selections here. And what we want to do now is save these selections from this acoustic file because let's say if after we had thoroughly analyzed this file, we're satisfied that we've collected all of our entries and we're now ready to report on this file. We will come up to our drop-down file menu in the upper left-hand corner, and we want to save this selection table. So save selection table, table number 1. It will default the name of this to the name of your file, the acoustic file that it is based on. You do not want to change the name of this file because then we will lose orientation to the original data file. So just leave this as its default file name, which will end in .table.1.selections.txt. These will be text files that are reported back to us as part of our citizen science project. So go ahead and save this file to your designated folder for your project. Let me find mine. Volunteer training, and I will save my text file. Okay, and once I have my text file, that is all that I need to report back to the Reef Sanctuary Soundscapes project. You will jump onto our website, find the data reporting link, and add that data to our project files for further analysis and observation by NOAA acousticians. We thank you so much for you participating with this project. This will help inform research managers so much about the life and the visitation at Gray's Reef National Marine Sanctuary. It will help inform us about protected species like North Atlantic right whales, when they may be present in our sanctuaries, when we may have visitors, vessel traffic, human noises of other kinds, as well as fish migration, spawning events, things of that nature, along with geologic processes or earthquakes and things of that matter. So there are a number of different research focus and issues and concerns and many other things to learn just by listening to the sea and being a part of the Gray's Reef Sanctuary Soundscapes project. So thank you so much for participating. We will have additional webinars, web trainings in the future to discuss these processes. In the meantime, if you have any questions, please reach out to us at graysreef@marinesanctuary.org.